We have seen about ETC cycle in previous class. In normal situations, in normal daylight, ETC cycle happens normally. But if there is excessive light, photosystems get damaged by the excessive light. How this happens, let us see. Due to excessive light, ETC cycle happens rapidly. So, proton ions get accumulated in the lumen rapidly. These excessive proton ions decreases the pH value. We already know that. Low pH means acidic, high pH means basic. Here, pH is getting decreased. So, this low pH creates acidity. Let us see how this happens. Due to the excessive light, singlet oxygen forms. Also, hydrogen peroxide forms. We already know that hydrogen peroxide is dangerous to the photosystems. These ions or these free radicals damages the photosystem. In today's lesson, we will see about how plant protects itself from excessive light. Watch the diagram. There is excessive light that fall on the photosystem too. ETC cycle happening rapidly. So, proton ions are increasing here. Overload happened on the photosystem too. So that singlet oxygen is formed. So that the main component of the photosystem too is D1 protein. That D1 protein is getting damaged. Peroxide getting increased here. So, what is the next step? DNA will be damaged and pigment degradation. The color will be vanished. What is the next step? In the ROS cycle, singlet oxygen forms, superoxide forms, also peroxide getting formed. What is the next step? Here already pigment degradation happened, so that it won't take further photons. So photoinhibition happens here. I explained everything about this diagram. Let us read out the theory part. Photosynthesis needs light, but too much light becomes dangerous. When light energy absorbed by chlorophyll exceeds the capacity of electron transport chain, Kelvin cycle, NADP plus reduction. So these all increases. Once excessive energy entered into the photosystem too, it cannot go out. This causes formation of reactive oxygen species. So this creates ROS, okay na? That is singlet oxygen, superoxide, hydrogen peroxide. Okay. This ROS can damage D1 protein of photosystem to lipid membranes, pigments, DNA. This light-induced inhibition called photoinhibition. So plants evolved photoprotective mechanism to safely dissipate extra energy. Let us see simply how this repair cycle happens. There is highlight. Damage happened to the D1 protein of the PS2. So here is the damage happened. So this should be removed. It will be removed by protease enzyme. So it is removed here. Now it should be synthesized and replaced by another new D1 protein. Okay. The next topic is non-photochemical quenching. We have discussed in first class. Okay. You might have remembered it. But let us see it in depth. There is highlight. pH value is getting decreased due to proton gradient is increasing, so acidity is increasing here. We have seen that D1 protein is damaged here, so it should be replaced with new one. The process happens with PSBS protein, okay? Please remember it. The main component that performs this action is zeaxanthin. So, zeaxanthin releases excessive light as heat. This is called heat dissipation. Let us come to theory part. Excess excitation energy is released as heat instead of photochemistry. This happens mainly in light harvesting complex 2, that is PS2. Okay. Let us see how this happens step by step. Step 1. Highlight increases the proton gradient. Strong ETC pumping. High proton. High pH value in the thylacyl lumen. Step 2. Xanthophyll cycle activation. Pigment changes from violaxanthin to anthraxanthin to zeaxanthin. Zeaxanthin helps dissipate energy safely. Step 3. PSBS protein sensing. PSBS senses low pH. That means acidity. PSBS senses here acidity. Conformational changes in antenna complexes. So PSBS identifies antenna changes. Okay. Step 4. Heat dissipation. Energy released as heat. Here excited chlorophyll relaxed without electron transfer. That means here plant is not solving the problem with electron transfer, but it is solving the problem with releasing the excessive light as heat. Types of NPQ. This is advanced concept. There is different NPQ systems in the plants. The first one is QE. energy dependent. It is fast. It happens in seconds or minutes. QT, state transitions, balancing PS1 and PS2. So it balances the electrons in PS1 and PS2. QI, photoinhibitory quenching. It slows the repair process. I told you earlier, if energy is more in PS2, that energy gets shared with PS1. Okay. Also, if PS1 have more energy, it shares with PS2. This is called state transitions. Okay. Let us see how this happens. Plants dynamically redistribute light energy between photosystems. When PS2 gets too much energy, LHC2 becomes phosphorylated. Some antenna move towards PS1. Energy balance restored. State 1. So this get balanced between state 1 and state 2. Purpose. Prevent overexcitation of PS2. So it prevents the damage of PS2 by sharing the electrons between two photosystems. Maintain optimal electron flow. So it keeps optimal between two photosystems. When PS2 gets too much energy, PS2 becomes phosphorylated. Some antenna move towards PS1. Energy balance restored. This process happens between state 1 and state 2. I told you in previous slide that PSBS protein recognizes the antenna molecule changes. So it is happening in state transitions. So PSBS identifies these changes. Let us go to next slide. The next topic is cyclic electron flow. If the Kelvin cycle has less energy, that means it does not have sufficient energy, this cycle happens. What happens here? If Kelvin cycle does not have enough energy, those electrons come back from the Kelvin cycle to the PS1. So those electrons create high proton gradient in the PS1. So this high proton creates proton gradient. This proton gradient supports the ATP synthase that combines the ADP plus inorganic phosphate and forms ATP. This ATP moves to Kelvin cycle. I think you understood now. Let us go to the theory part so that you can understand very clearly. When NADP plus is limited or Kelvin cycle slows, electron cycle back around PS1 instead of forming NADPH. So here NADPH is not forming but electrons are moving back. Why is this protective? Produces extra ATP, increases proton gradient, enhances NPQ heat dissipation, prevents over-reduction of ETC. Over-reduction means acidity is decreasing, means base is forming. So it prevents from over-reduction of ETC. That means it increases the acidity. Cyclic electron flow have two pathways, that is PGR5 or PGRL1 pathway or NDH pathway. Let us go to next slide. Now let us see fifth point, ROS detoxification systems. We know that. What is ROS? Superoxide, hydrogen peroxide, singlet oxygen. Okay. So they should be removed from the photosystems because they forms more acid. So they should be removed from the thylakoid. Okay. Let us come to the theory part. Even with prevention, some ROS still form. So prevention also won't stop the ROS formation. Plants use antioxidant systems. That means they are free radicals. Okay. Free radicals are removed by antioxidants. We know that. Let us see about it. We eat antioxidant foods. What are they? Vegetables and fruits. These antioxidant foods protects us from free radicals. Let us see how it happens. We eat yummy junk food outside. What is the junk food? We eat samosa, we eat pani puri, we eat bhaji or bhaji. Okay. We eat other fried items. These foods have free radicals. These free radicals damages our cells. What is free radicals? Free radicals means hydrogen peroxide. Okay. These molecules damages our cells. So they should be removed by antioxidants. Where antioxidant presents? Antioxidants present in vegetables or fruits. They are helpful in repairing our cells and keeps us healthy and also keeps us for living for more years. Let us come to the theory part. This process happens with enzymatic actions, that is superoxide dismutase, that is SOD, ascorbate peroxide, that is APX, and catalase. This also happens in non-enzymatic actions, that is ascorbate, that is vitamin C. Vitamin C includes antioxidants. We already know that. Glutathione, carotenoids. Carotenoids are present more in carrot. So eat carrot and have healthy cells. These forms the water cycle. Mehler reaction. Okay. Today's lesson is completed. If you like my videos, please subscribe my channel. I may try to finish 4 to 5 units until CSIR examination.